Hi everyone, welcome to CAPSA. Today we are going to learn about plasmids. So what is plasmid? A small piece of DNA, usually shaped like a circle, and it has two strands, just like normal DNA. Unlike the main chromosome of a cell, a plasmid lives separately, almost like an extra chromosomal part. Most plasmids are found in bacteria, but some appear in archaea and even in certain eukaryotic cells. A cell does not need a plasmid to stay alive, but a plasmid can give the cell helpful traits such as resistance to antibiotics or ability to use the special food sources. Because plasmids can copy themselves and sometimes move from one cell to another, they are extremely useful in genetic engineering and research. Let us look at the important parts of a plasmid so every name makes a sense. First is the origin of replication or ORI. This is the starting point where the plasmid begins copying itself inside the cell. ORI controls how many copies of the plasmid can exist. A strong ORI can make many copies, while a weaker one makes fewer. Next, we have the selectable marker. This is often a gene for antibiotic resistance. It helps us know that which cells have the plasmid. If we grow cells on a plate that contains that antibiotic, only the cells with plasmid will survive, making them easier to identify. Another key part is the multiple cloning site, sometimes called the polylinker. This is the short stretch of DNA filled with special cutting spots for enzymes. Scientists can open the plasmid at one of these sites and insert a new gene, almost like unzipping a bag to put something inside. Before the gene we insert, there is a promoter. The promoter is a short DNA sequence that tells the cell's RNA polymerase where to start the reading. It works like a switch that turns the gene on. Some promoters are always on, others can be turned on by certain signals, and some are designed for special host cells. When we insert our gene of interest, we place it so the promoter controls it. That gene could make a protein we want to study, a medicine, or even a colored or glowing signal. Many plasmids also have reporter genes like GFP, the green fluorescent protein. This allows us to see whether the plasmid is working because the cell glows under the light. Small primer binding sites are often built in so we can check the plasmid sequence quickly with the PCR. Some plasmids include extra features such as shuttle sequences that let the plasmid work in more than one species or small tags that help scientists collect and purify proteins made from the plasmid. Plasmids are made for different type of functions or jobs. Cloning vectors just make many copies of the gene. Expression vectors not only copy the gene but also make the protein from that gene. Shuttle vectors move easily between different species. Reporter vectors helps us study how active a promoter region is. Some plasmids are changed into viral vectors for gene therapy in the medicine. In nature, plasmids copy themselves on their own and have systems to make sure they pass into new cells when the bacteria divide. Plasmids can also spread from cell to cell in three main ways. Conjugation is a direct transfer through a small bridge called pilus. Transformation is when a cell picks up plasmid DNA floating in the environment. Transduction happens when a virus carries plasmid DNA from one cell to another. So, plasmids are important because their design is simple but powerful. They allow scientists to copy genes, produce proteins, test gene activity, and even deliver new genes into cells for therapy. By learning what each part does, ORI, selectable marker, cloning site, promoter, and the inserted gene, we see that a plasmid is like a flexible toolkit for the biology and also biotechnology. To wrap up, plasmids are not just tiny circles of DNA, they are key tools that connect basic genetics with real world uses, from making medicines to improving research. So, if this explanation helped you understand plasmids, please like this video and subscribe to the channel. Turn on the notifications so you will not miss next lesson. Together, we will keep exploring how life works at the molecular level. Thanks for watching.